Okay, welcome back everyone. How did you go with those tutorial activities? Those on campus, you will have done those in class. Those online, you'll have an opportunity to upload those tutorial answers so that you can compare them. If you have any questions, if you're online, just talk to your tutor. Okay, so what we just did previously, we started with the plasma membrane, and what we went through were the components of the plasma membrane. But in particular, the lipids, so that's your phospholipids, your cholesterol and your glycolipids, and also the proteins that are involved in the actual cell membrane itself. We then went through the functions of those plasma membranes, the plasma membrane proteins, and they can be either an integral protein or a peripheral protein. And there were 6 functions. Okay, do you remember the 6 functions? So we'll run through them very quickly. There was ion channel, which allows ions to go directly through. You then have carrier or transporters. They actually change shape, okay, so that the substance enters and then the protein changes shape and then puts it through to the other side. The next one we had, there's receptors. So receptor attached or linked up with a ligand, so a particular substance called a ligand attached to that receptor, and then what that receptor would do would be create an action within the cell. Sometimes the ligand goes through, sometimes it does not. The next one you had were enzymes. So enzymes, what they do is they catalyze reactions. So a substrate comes along and it makes the cell, it helps the cell go through its reaction. The next one was linker. So what these proteins act as, they help stabilize the cell membrane. So they attach or link together the filaments, which are part of your cytoskeleton. And then you had your self-identity markers. And that's where you need to identify, one cell needs to identify its own or your own body, okay? Where that's important is with transplants, organ transplants. Now, for those substances, or for substances to get across the membrane, the cell membrane, we need to look at how they are transported. Now, we lightly touched on, it was in that last section, about gradients. So when we're talking about gradients, we're talking about the particular concentration, be it a concentration gradient, and the particular charges, if it's an electrical gradient, on one side of the cell membrane to the other side. So the outside, which is this part here, so in this diagram, hang in there and I'll just draw it for you. So here, you can see that's the outside, and they are the ions that are on the outside. And then here you have the inside, so you have a concentration of ions here on the inside. The difference between the two, that is on the outside and what's on the inside, it creates the concentration gradient. Okay? So, if you have a look on the outside here, have a look at these ions. You've got little green circles, which are sodium, and you've got these little triangular type things that are potassium. Then you've got oxygen molecules and you've got carbon dioxide molecules. And then you've got same molecules on the inside. If you can see on the outside here, if you count up the number of sodiums, you've got 1, 2, 3, 4, 5, 6, 7, 8, 9. So you have 9 sodiums on the outside, and if you count the inside, you have 1. So there are more sodiums on the outside than the inside. If you count the potassiums, you've got 2 potassiums on the outside of the cell, and you have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 potassiums on the inside. So more on the inside than the outside. So generally, in a cell, sodium likes to be in higher concentrations on the outside, and potassium likes to be in higher concentrations on the inside. Okay, so the concentration gradient is the difference in those concentration between the two sides of the plasma membrane. An electrical gradient, on the other hand, is the difference in the electrical charges across the bilayer. So each of these ions, if you have a look, have a charge. So sodium's got a positive charge, potassium's got a positive charge. So the electrical gradient is the difference in the electrical charges across the bilayer. So what is the charge on the outside? So you can see that here. And what is the charge on the inside? Here. So you can see positive charge on the outside and a negative charge on the inside, depending on which ions are there. So it's the difference in the electrical charges across the bilayer. So the inner surface of the plasma membrane is more negatively charged, generally, and the outer surface is more positively charged. And what this is called is the membrane potential. It's like in a battery. So in a battery, you've got charges, you've got positive and negative. And that battery always maintains a charge so that when you turn the key to the car, it will turn on. So that is the potential, that's that membrane potential, which is the electrical gradient. Now the combined concentrations are called the electrochemical gradient. So now let's look at how things are transported across the membrane. Now there are three types of transport. You have passive transport, you have active transport, and you also have vesicular transport. Now vesicular transport will be covered in the next session. So this session we're only going to do passive and active. Okay, there are three types of passive transport. You've got simple diffusion, you've got facilitated diffusion, and that's divided into two types, channel and carrier. Have a think about where you've heard those words before, channel and carrier, in the last session, and that might give you a clue as to what is used to get things across. And the last type of passive transport is osmosis. When we're looking at active transport, we've got two types, primary and secondary transport. Okay, so you can see here, these are your different types here in the diagram. You've got simple diffusion, you've got channel-mediated facilitated diffusion, and you've got carrier-mediated facilitated diffusion. Osmosis, we'll look at a diagram later. Now you can see from the concentration gradient, there's going from a high concentration down to a low concentration. When we look at active transport, you've got two types. You've got primary, which is this top one here, and secondary here. And as I said, vesicular we'll look at further on down the track. I'll just get rid of this thing so we go back to our pointer. Okay, so passive transport. Definition. Okay, diffusion is the random mixing of particles, otherwise known as solute, that occurs in a solution, otherwise known as solvent, as a result of the kinetic energy of the particles. Particles are constantly moving, so they create their own energy from just moving around. Okay, so that's what it means by as a result of the kinetic energy. It's their own energy. They're not using anybody else's or anything else. It's just using its own energy. Now, molecules of the solute, which is the particles, and the solution diffuse from high to low concentration. So always in passive transport, which is to do with diffusion, okay, it moves from a high concentration to a low concentration. And if we go to the previous slide, you'll see from a high concentration to a low concentration. What factors can affect how fast particles or solutes, okay, diffuse across the plasma membrane depends upon certain things. So the first one is steepness of the concentration gradient. So the greater the steepness, the quicker the diffusion. So it happens faster. Temperature, the higher the temperature, the faster the diffusion. Now, size or mass of the diffusing substance, the smaller the size or mass of the substance, the faster it will diffuse across. Surface area, if the cell has a larger surface area, particles or solute will diffuse across quicker because you've got a larger area for it to go through. And diffusion distance, the greater the distance, the slower the diffusion. Okay, so you can see here in this diagram here, you have solvent, you have solute, which is this blue substance, pour the blue substance in, and eventually you reach equilibrium, which is balance. So it means that that solute is diffused evenly throughout the solution or solvent. Now, there's three different types, if you remember. The first type we're going to look at is simple diffusion. So the definition of simple diffusion, it's a passive process, so meaning it doesn't need any outside energy, it only uses its own kinetic energy, in which the solute moves freely through the lipid bilayer of the plasma membrane without the help of any membrane proteins. So remember, our membrane proteins, we had integral and peripheral, and they had 6 functions. In simple diffusion, it does not need a protein to get through. It goes straight through the plasma membrane. So if you have a look in this diagram here, you have a solute, a particle, that is going straight through the plasma membrane into the inside of the cell. Sorry, I'll go back. I'll keep clicking. Okay, so examples of particles or substances that transport through the plasma membrane by simple diffusion are non-polar, hydrophobic, so molecules that dislike water, such as respiratory gases, so you've got oxygen, carbon dioxide, nitrogen, some fats, so some fats will go straight through, small alcohols, okay, ammonia, and your fat-soluble vitamins, which are A, D, E, and K. They go through because they're fat-soluble, and your lipid bilayer is made up of fat, lipid. And some small uncharged polar molecules will go through, such as water, urea, which is a byproduct of protein metabolism, and some of your small alcohols. Okay, so that's simple diffusion. The next passive transport process is facilitated diffusion. Now, the definition of here is spontaneous passive process of solute, so particle movement across a biological membrane, which is your plasma membrane, via specific transmembrane integral proteins. So, okay, transmembrane, if you remember, goes completely from one side to the other. Now, the two types of passive transport are channel-mediated diffusion and carrier-mediated diffusion. So if you have a look up in the diagram here, you've got channel-mediated. If you remember, one of the 6 functions of your plasma proteins or plasma membrane proteins, one was acting as a ion channel. So channel-mediated facilitated diffusion utilizes ion channels, and it means that the particle can go straight through. And then you've got your carrier-mediated facilitated diffusion, and that is where they use a carrier or transporter, protein acting as a carrier or transporter. If you remember, with a carrier, particle goes in or solute goes in, the protein then changes shape, and then it can release it on the other side. So now let's go through each one individually. Okay, so channel-mediated facilitated diffusion. So a solute always moves down its concentration gradient. So as you can see here, okay, let's go here. So you can see it's going down its concentration gradient. So from a high concentration to a low concentration. The same as simple diffusion. So all of diffusion goes from a high concentration to a low concentration. But it utilizes or goes through a transmembrane protein acting as a ion channel. Now, the particles or the substances that it allows through, it allows inorganic ions, which are hydrophilic, so they like water because they're going from the outside to the inside, okay, and there's water in this channel, so they need to like water. So examples there are potassium, that's K+, sodium, which is Na2+, calcium, and chloride. Now, ion channels are very selective, and they're specific on the ion shape. So only a sodium can go through a channel that is an ion channel, a sodium ion channel. Only potassium can go through a potassium ion channel. Okay, so they're very selective. Now these channels can be gated or non-gated. So you can see here, wait till I find my little pointer. So you can see here it's got a little gate. Have a look at the little gate. Okay, so some of the channels do have gates. This one up here, you can see here, does not have a gate. It's not gated at all, but this one is. Okay, so that some may be gated and some are open all the time. So these ones here are open always. They can just go in and out as they please, but these ones here, the gated ones, will only allow the particle or the solute through when the gate is open, obviously. So the next type of facilitated diffusion is carrier mediated. So here, instead of using a channel, it uses a transmembrane protein acting as a carrier transport. Once again, to move the solute from the high concentration to the low, so down its concentration gradient. So the molecule, whatever it happens to be, binds to the carrier on one side, causes the carrier to undergo a change in shape, and the molecule can then be released on the other side. So you can see here it's changing shape to allow that molecule through. Okay, so once again, always from a high concentration to a low concentration, and it's facilitated. Why it's facilitated is because it's requiring something to help it. When we facilitate something, we give it assistance. Okay, so in this case it is a carrier mediated. Okay, this example here is glucose. So you can see glucose here comes and attaches to a protein called a glucose transporter, and it allows the glucose to enter the cell. So let's go into a bit more depth into the facilitated diffusion of glucose. So we've got here number one. Actually, hang in there, I'll just grab a pointer again. I'll grab a pen and I'll change colour. Let's go to a different colour. Let's go purple. Okay, so you can see here you've got the glucose. So this is your glucose here. You've got a glucose molecule here, and it binds, goes and attaches. You can see where it's attaching here. So it's just attaching here, okay, to a specific type of carrier transport protein called a GLUT, glucose transporter. So GLU, glucose, T for transporter, on the outside surface of the membrane. What happens then? You can see here it's changed shape, and the glucose passes through and is released into the, so here's your glucose molecule here, and it is released into the cytosol, into the inside of the cell. So that is one way glucose is transported into the cell. So you can imagine out here this would be glucose in the blood, because this extracellular fluid might be blood, and you need to get the glucose into the cell to provide energy for that cell. You don't want too much glucose in the blood, it needs to enter into the cell. So that is an example of facilitated carrier mediated diffusion. Hang in there and I'll just get rid of my pen. Okay, the last type of passive transport is osmosis. Now, its definition, it is the net movement of a solvent. Now, this is where it differs to the other two. The other two were net movement or movement of a solute, which is the particle. This is the net movement of the solvent, which is generally water, through a selectively permeable membrane, which is the cell membrane, from an area of higher water concentration to an area of lower water concentration, or from an area of low solute, okay, so whatever is dissolved in that water, the particle that's dissolved, to an area of high solute. So it is the movement of water, and it only occurs when the membrane is permeable to water, but not the solute itself. The solute doesn't move, the solvent moves, which is water, and it moves via simple diffusion is one way, and it can move through aquaporins. So there's two ways. It can move via simple diffusion, or it can move through aquaporins, which are integral membrane proteins that function as water channels. So have a look at this diagram. We've got this U-shaped tube. We've got water. So we've got little water molecules in here. Okay, we have a solute molecule, whatever that happens to be. It could be sodium. So we have all of these water molecules here on the left-hand side, and we have a selective permeable membrane, which may be our cell membrane. So what happens is the solvent, the water, needs to move from a high concentration of water molecules through to a low concentration of molecules. So hang in there, I'll just grab our pen again. Let's go to purple. I like purple. So you can see here you've got, it's the same level on both sides. So the water level is the same on both sides. Okay, at the moment. You've got all these little water molecules. So these are all these little dots here. And they have a high concentration. So there's a high concentration of water molecules here. Here's your semi-permeable membrane right here. And we know that in diffusion, particles, in this case water molecules, will move from, because it's the solvent, it will move from high concentration to low, because there's more little blue dots on this side than there is on this side. So the actual little dots, the water molecules, will move over to the right arm, where there was a lower concentration of water molecules. If you can see with the solute, okay, it is moving from an area of low solute, where there's no solute over here, to an area of high solute. But it's not the solute that moves, it is the water that moves. And it moves from a high concentration of water molecules to a low concentration of water molecules. It does that either through simple diffusion, or it uses little channels called aquaporins that are actually in the cell membrane itself. Okay, now a concept that you need to understand is tonicity. You might have heard of tonicity, and it's not to do with muscle tone. This is actually to do with a measure of a solution's ability to change the volume of a cell by altering its water concentration. We know that each cell in its cytosol contains about 55% water, actually in the cytosol itself. Excuse me. So we can have a solution on the outside of the cell that can affect the amount of water on the inside. And those three types of solution are isotonic, hypotonic, and hypertonic. You might have seen Gatorade or some of those types of rehydration drinks, and they say that it's isotonic or it's hypotonic. Or if you've been to hospital and had a drip, you'll find that the solution that's being hung up is either isotonic or hypotonic or hypertonic. So to explain each of them, in an isotonic, so if you've got an isotonic solution around the red, this is a red blood cell by the way, around the red blood cell, the cell maintains its shape and volume. So it means that the solute concentration is the same in the solution and the cell. Okay, so you can see water moves in and the same amount of water moves out. Same water moves in, same amount of water moves out. In a hypotonic solution, so if those red blood cells are floating in a hypotonic solution, the cell swells and it bursts, and that's called lysis. So in that case, the solute concentration is lower in the solution than in the cell, because we know, okay, that it moves from, what will happen is the solution or the water will move, more will move into the cell than out of the cell. So what happens then, if the cell fills with water, the cell will burst. So it swells with water, which will then, if it gets too big like a balloon, it will burst. Okay? So that's if there's a hypotonic solution around the cells. If there's a hypertonic solution around the cells, what happens there? The cells shrink, or it's called crenation. So what happens? The solute concentration, okay, in the solute is higher. There's a higher solute concentration in solution than in the cell. So what happens here? Water moves out. So more water moves out of the cell than stays in it. If you take water out of the cell will shrink. shrivel up and die. Okay, so that's tonicity. So we've done passive transport. Now we need to do active transport. So the definition of active transport, it's an energy requiring. So unlike passive, which use the kinetic energy of the cells themselves, their own energy, so their own energy created by movement, in active transport, it is requiring energy from somewhere else. Okay, so it's an energy requiring process that moves solute via carrier proteins. Remember what a carrier protein is? That's the protein that acts, or proteins that act as a carrier, where they change shape against the concentration gradient. So it goes from a low concentration to high concentration. So it is pumping it uphill, basically. It's like walking up a hill as compared to walking down a hill. Passive transport, down the hill, very easy. The momentum just takes you. Unlike up a hill, that's you actively need to use your muscles and a lot of energy to get up there, so you need some help, and that help is energy. There are two types of active transport. You have primary active transport and you have secondary active transport. Okay, so let's go through both of them. So primary active transport, it uses the energy derived from the hydrolysis of ATP. Now, do you remember which organelle produces ATP? If you remember, it's the mitochondria. So mitochondria produces ATP. So primary active transport needs the energy that's derived from breaking down that ATP. That energy then pumps a substance across the plasma membrane against its concentration gradient through a protein acting as a carrier or transporter protein. And these are called pumps. So the most prevalent is called the sodium-potassium pump. What it does, it maintains a low concentration of sodium in the cytosol, because if you remember, where did sodium like to be? Where was the greatest concentration of sodium? It was on the outside of the cell, and the lowest concentration was on the inside. Whereas in potassium, it likes to have a higher concentration inside the cell in the cytosol and a higher concentration on the outside of the cell. So what these pumps do, because we're going from a low concentration to a high concentration, okay, 3 sodiums are expelled into the extracellular fluid. Okay, hang on a minute, I'll get my pen again. Okay, ink color, we'll go red this time. Okay, so if you see here, we've got the sodium concentration gradient. So the normal concentration gradient, we have higher, so we have a higher concentration on the outside and we have a lower concentration on the inside. Sorry, that's not charge, that's actually concentration. Okay, so we have an increased concentration on the outside and a decreased concentration on the inside. With potassium, we have a increased concentration on the inside and a decreased or lower concentration on the outside. Okay, so what these pumps do, it pumps sodium against its concentration gradient. So it is actually pumping 3 sodiums out. So it's going against the concentration gradient through a pump called sodium-potassium ATPase pump. So the 3 sodiums, you can see, go from the inside and get expelled to the outside. How it does that is through the breakdown of ATP. So this ATP here has come from the mitochondria and it is broken down to ADP and P phosphate. So you start with adenosine tri, meaning 3. So there's 3 phosphates. Then it's broken down to adenosine diphosphate and a extra phosphate. Those two together make ADP. That reaction there of breaking it down is what creates the energy to allow the sodium to be pumped out. Now the other way, what happens is 2 potassiums are imported into the cell. We know that generally in potassium there is a low concentration here, and there is a higher concentration here. Okay, so what happens here? The potassium is pumped against its concentration gradient, so that two potassiums end up on the inside, so they are imported into the cell. So the sodiums are exported and the potassiums are imported, and that is against the concentration gradient. Requires energy through the hydrolysis or breakdown of ATP. Okay, the next one is secondary active transport. Okay, so what it is, the energy that is stored in the sodium or hydrogen concentration gradient drives other substances, so other than sodium or hydrogen, against their own concentration gradient through, once again, through carrier or proteins that act as carriers. They're called co-transporters. Now that gradient, so that sodium or hydrogen concentration gradient, is established by the primary active transport. So when that active transport happens, it creates a concentration gradient. Okay, and the energy is stored in either a sodium or a hydrogen concentration gradient. What that does, it allows sodium to leak back into the cell. So with secondary active transport, there are two types of proteins or carrier proteins. There are antiporters and there are symporters. Now, antiporters, the carrier protein moves two substances in the opposite direction. So we know that sodium, so we've got here, sodium comes back into the cell. It was pumped out, now it was pumped out by primary active, now it needs to come back into the cell. So you can see here is sodium coming back into the cell, it's leaking back in, because that's the way it naturally goes from high concentration to low concentration. But now we need to pump calcium out of the cell against its concentration gradient. And it does that through a carrier protein called an antiporter. Anti, one going one way, the other going the other way. Okay? Symporters, on the other hand, move two substances in the same direction. So what we've got here, once again, we've got sodium leaking back through. Okay, so sodium's coming back through with its concentration gradient. Okay, and we also need to bring in glucose into the cell, because that's the way that glucose likes to go. Okay, but it needs a carrier protein to do it. So it's bringing in two substances in the same direction. So an example there, you've got glucose, and another example here is your amino acid. Okay, so antiporters, two substances in the opposite direction. Symporters, two substances in the same direction. The energy, where they get the energy from, though, is from the energy that was created or stored as a response, or what ultimately the primary active transport did. So let's go back very quickly to primary. Remember, it was the energy derived from the hydrolysis. So what happens when ATP is broken down? It creates a concentration gradient, okay, that is stored in sodium and hydrogen. So that energy that was produced by the breakdown of ATP to ADP and P, that energy is stored in a concentration gradient, and that what is used to drive substances across by secondary active transport. Okay? Wait till I just get my pointer back. Okay, so let's go through the example here. So the sodium glucose symporter. So we know symporter moves two things in the same direction. We know that the normal sodium gradient is from high to low, okay, from the outside to the inside. So primary active transport via the sodium-potassium pump drives an increase of sodium to the outside. So what happens here? And there we just get my coloured pointer. Sorry about there's no easy way to do this. Okay, so we've got here, okay, normal gradient is more sodium on the outside and less on the inside. What happened though, as a result of the primary active transport, more sodium ended up on the, okay, it pumped it to the outside. So what happened is it pumped sodium to the outside as a result of the primary active transport. So now we have a lot of sodium on the outside here. So more sodium on the outside than the inside. So now what happens, because you've got more sodium on the outside than the inside, that sodium then can travel down its concentration gradient, which is this here. So down the concentration gradient into the cell via the symporter. So this is the symporter here. What also happens as that sodium enters, so does glucose. So it, through the carrier protein, enters the cell as well. So now we have sodium coming in and we have glucose coming in. Okay? And that is the sodium glucose symporter. An example of an antiporter is a sodium calcium antiporter. So primary active transport via the pump once again drives sodium outside the cell. So initially sodium gets pumped out of the cell. What happens then with the energy that's stored in the sodium or hydrogen concentration gradient, sodium then enters the cell, because that's its natural way of going. That's what it normally would do. Okay, it would normally enter into the cell, so it leaks back in, and it enters via a antiporter. And if you remember, an antiporter is something that moves things in the opposite direction. So then what happens is the calcium then moves out of the cell, okay, against its concentration gradient. So now with this, the sodium is with its concentration gradient and the calcium is against its concentration gradient. Okay, because it's moving two substances in different directions, that's why it's called a antiporter. Okay, wait till I just get my pointer back. Okay, wait till we just get my pointer back. Okay, so we've come to the end of this session. Very big session, but transport is a very big session. Okay, so recap of the plasma membrane structure. So you need to come back to this diagram and have a look at the structure of your lipids, which are your phospholipids, your cholesterol and your glycolipids, and also your proteins, which are your integral and your peripheral proteins. You also need to, so that's very important that you do that. You also need to go then through the functions of the plasma proteins, the 6 functions of the plasma proteins, and then you need to go through the transport, or how things get across the plasma membrane. 2 types that we did this session, and that was active and passive. 3 types of passive, which was simple, facilitated, which is divided into channel mediated and carrier mediated, and also osmosis. And then your active, you have secondary and primary. Okay, so preparation for next session, you need to read chapter 3, and you need to talk about transport in vesicles, because we're going to do about transport across the plasma membrane, the 3rd type of transport, which is vesicular transport, and also review your active transport process, because vesicular transport requires active transport process. You can also, now what you can do is go to your tutorial and you can complete activity 3 and activity 4. So very important that you do those, making sure that you take notice of your diagrams that are in the tutorials, because they're very important diagrams to make sure that you can understand them and label them. And then also you need to complete the possible exam questions. Okay, so those possible exam questions, once again, could be on the final exam. On campus, you'll be able to do them in class and get into groups and do the answers. Online, in your groups online, you will can do your answers, and you can, within your tutorial group, you can compare your answers, or you each can do one or whatever. That's up to you, and your online tutor will give you assistance with that. Okay, so that's it. I will see you next time. Bye.